Hello dear students, welcome back to our YouTube channel FoodTech Online. This MCQs are related to ICAR-PG Food Science and Technology. Today we will be covering set number 8 MCQ. So let's begin. We have also launched an ebook which is beneficial for the ICAR-PG Food Science and Technology students, which covers all the important features, and the core subjects are food chemistry and nutrition, food microbiology, food products technology, food engineering, and the link of this ebook is given in the description box. Please have a look at it. Question number 1. Which of the following is known as radiation sterilization? Options are radapatization, radicidation, radiorization. Option D is none of the above. And the correct answer is option number A, radapatization. Now, as the name suggests, it is the radiation. It is the radiation apatization. And apatization also means sterilization. That's why it is very easy to remember that it is radapatization, that is radiation sterilization. Now, what is food irradiation? That we have to know basically. So, food irradiation uses high-speed electron beams. So, food irradiation uses high-speed electron beams. And they are in the wavelength of 200 to 2000 angstroms. Okay, so the examples of these can be the X-rays, gamma rays, and these are having sufficient energy to break the chemical bonds and ionize the molecule, and thus they will kill the cellular membrane, or they will change the structure of the cellular membrane inside a microbial structure. That's why they inhibit their growth. So depending upon the dosage, now the dosage is given in the form of either the grayscale, which is written as GY, and it is also measured in joule per kg. So 1 gray is equal to 1 joule of energy absorbed by 1 kg of the food. 1 kg of the food. This is also equal to 100 radiations or 100 rad. So depending upon the dosage, there are three categories. One is the radapatization, second is the radicidation, and third is the radiorization. So the radapatization is also known as radiation sterilization, which uses the grayscale from 25 to 45 kilogray. So this uses 25 to 45 kilogray of the radiations. The radicidation uses 2 to 8 kilogray, and the radiorization uses 0.4 to 10 kilogray. Question number 2. Which of the following is equivalent to pasteurization? Options are radapatization, radicidation, radiorization, and option D is both A and B. And the correct option is option number B, radicidation. So radicidation uses 2 to 8 kilogray of the radiation, and it will kill the specific non-spore forming pathogenic bacteria and except some viruses. It will reduce the number of the viable specific non-spore forming pathogenic bacteria, and thus it is equivalent to pasteurization and not sterilization. So here our correct option is radicidation is equivalent to pasteurization. Question number 3. Which of the following is equivalent to irradiation? Options are option A, radapatization, B is drying, C is heating, option D is all of the above. And the correct option is option number A, radapatization. So the irradiation, it is also called as cold sterilization or pasteurization. So the irradiation is a method which uses high speed of electron beams. It can be the X-rays, gamma rays, which have the sufficient energy to break the chemical bond and ionize molecules, and they also act as a germicidal, as it destroys or change the structure of cellular membrane and damage the DNA and RNA, and thus it will inhibit the growth of the microbes. The irradiation or the high electron beams are generated using cobalt 60 as a source of energy and cesium 137. So the unit which is the unit of absorbed dosage is denoted by gray, and this irradiation process will destroy the microorganisms, or it will inhibit the ripening, it will inhibit the sprouting without affecting the flavor, without affecting the flavor and structural changes in food. So here our correct option will be radapatization, that is radiation appetization or radiation sterilization. Question number 4, which is true about UV rays. UV rays is ultraviolet rays. Options are wavelength is 2000 Armstrong unit. It is very much capable of destroying harmful microbes. It is non-ionizing. Option D is all of the above. And the correct option is option number D, all of the above. So the ionizing radiations are, there are three types of ionizing radiations, which are the alpha, beta, and gamma, while the non-ionizing are the ones like the UV radiation, ultraviolet rays. Then we have got the visible, then this is the visible rays are also non-ionizing. Then IR is the infrared, and then microwave radiations, and last one is the radio frequency, which is the RF. So these are non-ionizing radiations, and these are having a range of 10,000. These non-ionizing radiations are in the range of 10 to 4000 Armstrong units. So Armstrong is written like this, Armstrong unit. So here the both options are right, that is the wavelength is the wavelength is 2000 Armstrong unit, and it is also non-ionizing. Also UV benefits in food as it kills the bacteria without the heat or harsh chemicals. Thus it is also capable of destroying harmful microorganisms. So in this case all the three options are correct. That's why correct option is all of the above. Question number 5, which rays have more penetrating power? Options are cathode rays, beta rays, gamma rays, and the UV rays. And the correct option is option number C, the gamma rays. So what is penetrating power? Penetrating power is the ability of a ray to travel through a material. So it is ability to travel through a material. For example, the gamma rays, which are having the higher penetrating power, these can penetrate through several meters in a dense material like the lead or concrete. And the penetrating power is inversely proportional. Power is inversely proportional to the wavelength. So if it is the shorter wavelength, shorter wavelength will have more penetrating power. So for example, the alpha particles, the beta particles, these are having smaller mass and they have high ionizing energy. Thus, they cannot penetrate through any material very far. For example, the gamma rays, which doesn't have any charge or the mass, it has the least ionizing energy and thus has the highest penetrating power. So here our correct option is option number C, gamma rays. Question number 6. In which year did the Prevention of Food Adulteration Act pass? This is also called as PFA Act, Prevention of Food Adulteration Act. This was options are 29 September 1954, 1955, 1953, 1952. And the correct option is option number A, 29 September 1954. So what is the PFA Act? It is the Prevention of Food Adulteration Act, which was aimed to the aims of this act are firstly to prevent adulteration of the foods, prevent adulteration of food. Also, it will sale of prevent sale of adulterated food. Thirdly, to protect public protect public from unsafe food, unsafe or adulterated food. And the fourth one is to eliminate fraudulent practice. Eliminate fraudulent practices. That is unsafe methods of production of the food or any adulteration is being added, such type of practices has to be eliminated. So under this act we have the prevention of adulteration of food. It will prevent the sale of substandard or the adulterated food. It will also protect the public from the unsafe food by providing awareness, and the last one is to eliminate the fraudulent practices. The adulteration of food is the um, addition of or it is alteration of food quality done deliberately with the aim of economic advantage that will change the properties of food, including its color, texture, weight, taste, volume and shelf life, etc., and the food will fail to meet the legal standards. So that is called as adulteration. So here our correct option is 29 September 1954. Question number 7. According to the Indian government, how many groups have been recommended for irradiation? Options are 10 groups, 12 groups, 8 groups, 14 groups. Correct option is option number D, 14 groups. Question number 8. Why is irradiation important in garlic and onion? Options are to prevent rotting, to prevent germination, to prevent post-harvest diseases, to prevent post-harvest insect. And the correct option is option number B, to prevent the germination. So irradiation is a process which involves the exposing of the food products to a controlled amount of ionizing radiation or the high electron beams, and this treatment is carefully calibrated to prevent the uh, germination or the sprouting, and also it will arrest the growth of the microbes. So why it is important in garlic and onion? This is to prevent germination. Now how it prevents the germination? Firstly, it damages the cells which are present inside any microbes. So it damages the cell division process. Secondly, it will inhibit the enzyme which are inhibit enzymes responsible for sprouting or germination. And thirdly, it will reduce the respiration rate. Reduce respiration. So if the respiration rate is reduced, then the water or the moisture will not be generated, and thus it will prevent the sprouting. So it will slow down the metabolic processes and thus reducing the energy available for sprouting. So here our correct option is to prevent the germination. Question number 9. ISOMT, which is the irradiation sterilization of medical products, is located in options are Bangalore, Delhi, Mumbai and Thiruvananthapuram. Correct answer is option number C, Mumbai. So it is located in the Chembur district of Mumbai or Maharashtra, and it is a facility under BARC, that is Bhabha Atomic Research Institute, Bhabha Atomic Research Center, and it is a gamma radiation processing facility for healthcare products. For healthcare products. So it was established in 1974 under the Department of Atomic Energy. Question number 10. Which is the principle of radiation? Options are to increase the shelf life of food, to prevent the fruits from ripening, to kill the harmful microbes, and option D is all of the above. And the correct option is option number D, all of the above. So how the radiation prevents the fruits from ripening is it will degrade the starch, and it will prevent the formation of degrade starch and prevent formation of fructose, which is a fruit sugar, and glucose. So once the starch is degraded, it will prevent the formation of both, that is the fructose and glucose, and it will prevent the fruits from ripening. Secondly, how it will increase the shelf life of the food products? The ionizing radiation breaks the chemical bond. It will break chemical bond, and it will disrupt structure, that is DNA and RNA. It will disturb both these structures, so the internal structure of the cell membrane of the microbes will be destroyed, and thus it will kill the microorganisms and it will eventually increase the shelf life of the food products. It will also inhibit some of the enzymes which lead to the sprouting or which can cause some chemical reactions. So altogether it will prevent the fruits from ripening or sprouting, it will inhibit the enzyme action, it will kill the microorganisms by disrupting the DNA and RNA from the cell structure, and altogether factors will contribute to the increase in the shelf life of the food product. So here the answer is all of the above. Question number 11. What is the lethal dose of ionizing radiation in fusarium? Options are 2,50,000, 15,000, 10,000 and 5,000. Correct option is option number A, 2,50,000. So conversion is 1 kilogram is equal to 1 lakh. So the minimum lethal dose for fungi or any type of mold is minimum is 1.5 kilogram and above. This is the minimum one. So anything above this will kill the mold or the fungi. So here we have the organism which is fusarium, which is a fungi. So correct option will be 2,50,000 because this is given in rats. So it can be 2.5 kilogram. Question number 12. What is the water activity in fruits and vegetables? Options are necessary moisture content in fruits, flow of water inside the foods, required moisture content for the microorganisms. Option D is none of the above. And the correct option is option number C, required moisture content of microorganisms. So the water activity is also denoted as AW. So it is defined as the ratio of water vapor. So water activity is the water vapor of food divided by water vapor of the pure water. So it is the ratio of vapor pressure inside the food. It is also denoted by the P and divided by the vapor pressure of pure water at P0 or the P initial at same temperature. At same temperature. So it measures the activity, uh, it measures the free and unbound water present inside the food. So it measures the free and unbound moisture in food. So it is that available moisture which can be then taken up by the microorganisms to grow. So depending upon the microorganism, the water activity can vary. So some type of microorganism like the bacteria will require a higher water activity, that is above 0.86, and the yeast and mold require less water activity, that is 0.65. So if the water activity of the food is below 0.5, then most of the microorganisms won't grow. Question number 13. What is the formula for water activity? Options are W is equal to P by P1, W is equal to P1 by P, W is equal to P1 by P into 100, W is equal to P by P1 into 100. So correct option is option number A, W is equal to P upon P1. So water activity can also be written like AW, and it is the ratio of vapor pressure of the food component divided by vapor pressure of pure water, both at same temperature. So this can be denoted by P and this can be denoted by P0 or the absolute or the initial. So it can be denoted by P1 or P0. So this is the formula. Question number 14. What is the relation of water activity with relative humidity? Options are water activity is equal to relative humidity. This can also be denoted by RH, and water activity as AW. So RH into 100, water activity is RH divided by 100, or relative humidity is equal to water activity into 100 or water activity divided by 100. So correct option is option number B, water activity is equal to relative humidity by 100. So what is water activity? It is the amount or the availability of water for chemical and biological reactions which is present in the food. So water activity is the unbound moisture, unbound moisture in food, unbound moisture in food, whereas the relative humidity is the amount of water vapor in the air compared to the maximum capacity. So it is expressed as the percentage of maximum humidity at a given temperature. So the units are, the water activity is measured in the scale of 0 to 1, whereas the relative humidity is measured in percentage and it ranges from 0 to 100. So that is the saturation. Now the relationship between us, water activity is relative humidity divided by 100. So for example, if the relative humidity, example, if the relative humidity is 60%, then water activity will be 0.6. So this relationship is important because it will help us to control the water activity as well as relative humidity, and it is crucial for preventing the microbial growth, corrosion and other processes. Question number 15. What is the water activity of skim milk powder? Options are 0.87 to 0.8, 0.75 to 0.6, 0.95 to 0.91. Option D is 1 to 0.9. Correct option is option number B, 0.75 to 6.6. So the milk powders are considered low water activity foods with the typical water activity values which are less than 0.7. These are having very low moisture, hence most of the products are not affected by microbes, especially mold, bacteria, yeast or fungi. Question number 16. What is true about the normal drying curve? Options are: drying is faster in later stage, drying is slow in the later stage, drying is too fast in the later stage, drying is very slow in the later stage. Correct option is option number B, drying is slow in the later stage. So this is a drying curve and it is plotted against the drying rate which is represented as kg per meter square versus the water content represented as kg of water per kg of solid per kg of dry solid. So the initially this is the moisture content, then it will have a constant falling rate period, then it will have a first falling rate period, and then is the second falling rate period. So this is A, B, C, D and E. This point is E. So this point is called as critical moisture content. Critical moisture content. And the drying rates are B to C is the constant falling rate period, followed by C to D is first falling rate period, and this is the second falling rate period. So initially the drying of the food material is faster because from point A to C the only surface, so from point A to C surface moisture or the unbound or free moisture is removed. Free moisture removed. And then the falling rate period are divided into two groups, that is C to D is the first falling rate period and D to E is the second falling rate period, where the unbound moisture is removed. So it will take a longer time to dry because the unbound moisture have to travel from the internal structure of the food to the surface and then it will get dried. So this is the drying curve, and here our correct option is drying is slow in the later stages. Question number 17. Which factor affects the drying? Options are atmospheric factors, nature and constitution of the drying material, both A and B, and last option is none of the above. Correct option is option number C, both A and B. So the atmospheric factors like the temperature, humidity, and the nature and constituent of the drying material. Nature means if it is acidic, basic, or any type of the chemical reaction. Constitution means how it is packed, like what is its structure, crystalline or amorphous. So depending upon the nature and constitution of the drying material as well as the atmospheric factors, drying will be affected. Because if we increase the temperature and lower the humidity, drying will be faster. If uh, if a material is more amorphous, it will have a very porous structure and the moisture will be removed faster. And also it will depend upon the other factors like the surface area. So if more surface area is exposed, more water will be or more moisture will be removed. So these are the factors which affect the drying. So here our correct option is option number C, both A and B. Question number 18. What is the temperature of air inside a bell tripe? Options are 100 degrees Celsius, 135 degrees Celsius, 110 degrees Celsius and 109 degrees Celsius. Correct option is option number B, 135 degrees Celsius. Question number 19. Which is an ideal dryer for drying high quality orange juice? Options are continuous vacuum belt dryer, vacuum shelf dryer. Option C is freeze dryer and option D is home scale dryer. And the correct option is option number A, continuous vacuum belt dryer. So fruit juices, especially the any type of fruit juices will be high in sugar, and especially in case of orange juice, it will be high in vitamin C, high in vitamin C or the ascorbic acid. And when we are applying high heat, so if we apply, if the temperature is more, if temperature is higher, the sugar can char or it will get caramelized. Sugar can char and vitamin C is lost during the high temperature processing. So the better method of preparing orange juice, high quality orange juice is using a continuous vacuum belt dryer, because when we apply vacuum, the temperature or the boiling point is lowered down. Boiling point decreases when we apply vacuum. So it will preserve the flavor, aroma, as well as the vitamin C content will not be lost as much as if we apply the high heat. Okay, so freeze drying can also be a technique for drying, but after freezing, it will then has to be applied, a vacuum has to be applied so that it will evaporate the moisture. So the best option is a continuous vacuum belt dryer. So what happens? It will gently remove the water from the juice using a vacuum, and slow moving belt will preserve the juice's flavor and nutrition. So vacuum shelf dryer, again what will happen if we are using shelf dryer, the juice will stay in one place, juice is not moving, so so the temperature which is applied will be at a localized point. So continuous motion of the uh, juice is necessary. So continuous vacuum belt dryer is the best option. Question number 20, which is true about the ionizing radiation? Options are: wavelength is more than 2000 Armstrong unit, wavelength is 2000 Armstrong unit, and wavelength is between the range of 2000 less than 2000 to up to 2000 Armstrong unit. Option D is any of the above, and the correct option is option number C. The wavelength is less than 2000 or 2000 Armstrong units. So the radiation can vary from 10 to 2000 Armstrong units. Question number 21. Which of the following is a method of preservation? Options are drying, canning, freezing, all of the above. Correct answer is option D, all of the above. So we can extend the shelf life of the food using various techniques of preservation, and it can include the drying technique where we apply heat at optimum temperature. So the drying can be sun drying or mechanical drying. There are different types of drying, sun drying or mechanical drying. So the concept behind this is to reduce the moisture, to reduce moisture, so it won't be available for the microorganism to take up and grow. So once we reduce the moisture by drying, the food will be preserved. Second is the canning process, where we apply the heat and sometimes pressure so that the spores or spore forming microorganisms can also be killed. The canning temperatures can vary from 100 degrees Celsius to 121 degrees Celsius, and depending upon the pressure to be applied or not, even the pressure conditions can vary. It can be at sea level, the pressure can be up to 10 to 15 psi, that is pounds per square inch. So this will give a sterile environment, because food product is packed inside a sterile container, and then once it is exhausted, then it will be exposed to high temperature and pressure conditions, and the steam will form a vacuum inside the cans, so there won't be any contamination. So canning, the canned products can last up to 1 year, so it will have a longer shelf life. Third is the freezing technique. This won't kill the microorganisms, but it will inhibit the growth of enzymes and microorganisms that can spoil the food. Enzymes and microbes that can spoil the food. So it will only inhibit or arrest the growth of enzymes and microorganisms, but still it is one of the preservation techniques, because if we maintain the freezing conditions, that is below -18 degrees Celsius or still below it, that is up to -30 degrees Celsius, the food will be preserved for a longer period of time. So here our correct option is all of the above. Question number 22. Which one is the key requirement of food preservation? Options are: for future need, to preserve the food when the availability of food is more than the present use, for more marketing, and option D is all of the above. Correct option is option number B, to preserve the food when the availability of food is more than the present use. So for future need is also one of the requirement of food preservation, but the key requirement is the second option. So what happens when the season is there, mostly of the fruits and vegetables, they are grown in ample quantity. Seasonal fruits and vegetables are grown in ample quantity, so it can go to waste. So to reduce the waste, if all the lot is not consumed raw, then this can go to waste. So we apply preservation techniques. The preservation techniques can be freezing, it can be drying, or it can be a use of chemicals or the chemical preservatives to extend the shelf life of the food, so that it will be also available for us for the future need. But the basic or the key requirement is to reduce the wastage and to preserve the food when it is available at a ample quantity. Question number 23. What is canning? Options are: application of high temperature to food, application of low temperature to food, both A and B. Option D is none of the above. Correct option is option number A, application of high temperature of food. Canning is a food preservation process. It is a food preservation process, and it involves the packaging of the food into an airtight container such as the tin cans or the glass jars, and then heating them to a high temperature to kill any spore forming or any type of bacteria or microbes. Sometimes the pressure is also applied. Uh, Depending upon the pressure applied or not, it will kill the spores if there are present of any type of microorganism. So this process is also known as retorting or apodization, and it creates a vacuum seal that prevents the new contaminants from entering the container. So it will preserve the food by killing off the harmful bacteria or the pathogenic microorganism. It will prevent the entry of the contaminants, and it will also create a sterile environment, and thus the shelf life of canning is higher. So it has got higher shelf life. The shelf life can be up to an year. So depending upon the methods, like there are two methods of canning. One is the water bath canning. So this is done at a temperature around 100 degrees Celsius at sea level, and no pressure applied. No pressure. Second one is the pressure canning, where a temperature of around 121 degrees Celsius is applied at a pressure of 10 to 15 psi, that is pound per square inch. This type pressure is applied at sea level. So if we increase the temperature and the pressure conditions, it will remove most of the spore forming or the spores of the microorganism. The water bath will only kill the viable pathogenic microorganisms. Question number 24. The gluten formation started with stage of bread making. Options are hydration, development, proofing, and baking. Correct option is option number A, hydration. So gluten is a protein which comprises of two components, that is glutenin and gliadin, and these two after hydration will form gluten, which will give a viscoelastic nature, viscoelastic nature to the dough which is formed. So hydration is important because after hydration, these two, the starch molecules will puff up and proteins will form a network. So during the hydration stage, the gluten will start developing. So it will develop properly during the development stage, or where the kneading or mixing is done. Kneading or mixing, it will develop fully. Gluten develops. And why gluten is necessary? Because it will trap the gluten is necessary to trap the carbon dioxide and steam or the water vapor which are released during baking process. So this will give the structure or rise to the bread or any type of bakery products. So to trap this carbon dioxide and steam, the gluten network is a strong and sturdy gluten network is necessary. So here our correct option is hydration. Question number 25. Food sterilization via irradiation is also known as. Options are gas sterilization, cold sterilization, neutral sterilization, and all. Correct option is option number B, cold sterilization. It is called as cold sterilization because it does not raise the temperature during irradiation. So there is no type of heat involved. It is directly the high electron beams or the rays which are passed through a food product, and it doesn't raise the temperature. It will directly pass through the cellular structures of the microbes, and it will kill the microorganism. It will inhibit the sprouting and ripening. So it is also called as cold sterilization because it kills most of the microorganisms. So here our correct option is cold sterilization. So we have come to an end of this today's MCQ session. Please go through our app, that is FoodTech Online. It covers all the necessary information regarding most of the exams, that is FSSAI, GATE and state FAOs. It covers most of the subjects like the food processing, food chemistry, food engineering, etc. It has got various modules. It will definitely help you with the exam preparation. So the link of this is given in the description box. Please join our app. Also, do subscribe to our YouTube channel, FoodTech Online, and join us on the social media. And please like and share this video. Thank you.